you, and hello everyone. I'm excited to share with you some information about raw materials and process optimization that is important for drug developers and process development scientists in the mRNA therapeutic space. Let's start by going over some learning objectives. First, we will discuss the importance of raw materials in mRNA production. After that, we'll discuss the critical quality attributes of mRNA materials from the perspective of the manufacturing laboratory. Finally, we will dive into some mRNA synthesis and scale-up considerations with a focus on parameters for high mRNA yield. First, let's go over the importance of raw materials. This first slide shows the flow of drug development from target development all the way to commercial approval. Each phase has its own set of steps, varying scale, and different levels of consideration when it comes to raw material and manufacturing. In animal studies, the goal is to find a drug target quickly, which requires raw material to be flexible and at small scale. Once a drug candidate is identified, drug development works from small to larger scale to identify process parameters that will generate a consistent yield and quality from batch to batch under GMP guidelines for the manufacture of a drug candidate. Once a process is locked, the drug manufacture phase makes a drug at scale, focusing on process and product consistency. Thus, decisions concerning raw materials should be made well before the manufacturing phase. And the arrows in this slide indicate where decision points about material quality needs to be made. And decisions at these steps are critical for the success of your product. So what is an example of a scenario where product quality is not critically considered? This slide tells the story of a company working on a SARS-CoV-2 vaccine that followed the classical pathway of getting a C of A, asking if the material is animal origin free, and then getting a quote. They chose the cheapest material. In this case, it was a restriction enzyme. They made mRNA and tried to get it through regulatory agencies. The regulatory agencies asked for more documentation on the origin of the restriction enzyme since it was involved in the process. The company didn't have it because the material supplier didn't provide that level of documentation for the product. In desperation, they asked all other suppliers of that enzyme for the documentation, and no one had it. So they couldn't use the mRNA they had made with this enzyme to start their clinical trial. Because of the choice to go with the lowest cost vendor, the company's project was stalled, and other pharma companies passed them to produce a COVID vaccine. They had to remake the material, which costed hundreds of thousands of dollars. They ultimately lost over $300 million because of a raw material decision made on price, thinking that everything else was the same. But when they bought their material, of course, it was to save money. In this case, the product quality attributes weren't sufficient to stand up to regulatory scrutiny. The message of this story is that if you think all materials are made the same, and you make a choice primarily based on price, you're putting yourself at some risk. All materials aren't the same. So you need to ask questions and make sure you have the full quality picture for any material you choose. So we have 4 goals we recommend customers follow when selecting raw materials. The first goal is quality, which is at the core of every decision that is made to ensure risk is reduced as much as possible, which is our second goal. Risk can come from several sources: documentation, analysis, run-to-run yield variability, failure rate, and of course, supply chain risks. Supply chain risks are reduced by ensuring you have a secure material supply at the same quality level in the process development and manufacturing phases. Finally, start with the end in mind. That means to think about what the final process is going to look like at full scale from a procedural, parameter, and materials perspective. Keeping these 4 goals in mind ensures a smooth, low-risk process. So if quality is first and foremost, let's dive a little more into the critical material quality attributes for manufacturing mRNA. The list shown above highlights some critical quality attributes for GMP material. First, the manufacturer of material should follow ICH Q7 principles. Ensure the material manufacturer you work with follows those guidelines along with the process and end product that is animal origin free and beta-lactam free. In addition, You will want validated manufacturing processes and analytical methods along with verified compendial test methods. The materials should be tested for material-specific stability data and have detailed impurity profiles to ensure the products will last you a long time. Finally, all the quality support documentation should be available on demand. And what are those quality documents? The quality documentation needed for GMP manufacturing of mRNA therapies is listed above. In addition to these files, Drug Master Files, or DMFs, can really help simplify and speed up the process of submitting a new drug to the regulatory agencies. The DMF is on file with the regulatory agencies and contains comprehensive quality information about the products. Customers should work with their material distributor to get access to the DMF that they then can reference when submitting their drug for approval. So in addition to having well-documented products that meet key quality attributes, the process for material synthesis should be validated. So what is validation and how does it compare to a verified process? This slide shows an HPLC chromatogram analysis of N1-methyl-pseudo-UTP. Both were made by the same process. But the lots of material on the left were made by a verified process, while lots of material on the right were made by a validated process. Every lot of material passed our QC specs. While the product is similar and of good analytical quality when made by either process, the product made by a verified process has some issues. Different lots show different impurity levels of the same impurity. Some lots of material have different impurities from other lots, and the impurity profiles are not consistent from lot to lot. The inconsistency of a verified material is problematic and could cause delays of production due to required material inspections, lack of reproducibility, and the possibility of rejection due to new impurities. In contrast, the product made with a validated process is very consistent from lot to lot and does not display the variability seen in the material made by a verified process. This makes it much easier and less risky to use material like this in your manufacturing process. In addition to having a validated process, that products should be characterized and individual impurities should be quantified. This slide shows HPLC chromatograms of ATP, CTP, and GTP. The tables contain the identity and amounts of impurities found in our NTPs. The key highlights are these NTPs are all made with validated processes. None of these impurities are critical and could potentially interfere with mRNA synthesis. And each lot of NTP produced is consistent and predictable in terms of product quality. So with characterized and quantified impurities, you will know the material has met strict quality acceptance criteria prior to release. And now that we have a foundation into the critical material aspects for IVT, let's shift into some process development work that was recently done to optimize IVT process parameters. That way you will gain information on how to improve your IVT from both a quality and now process perspective. In order to optimize parameters for IVT or in vitro transcription, the R&D department within Thermo Fisher Scientific conducted a design of experiment, or DOE, to assess multiple parameters in the IVT process and see how those parameters interact. The parameters were buffer type, buffer concentration, nucleotide concentration, enzyme cofactors like magnesium and spermidine, and pH. The measure of response for the IVT was mRNA yield and mRNA quality. This slide summarizes the data from that DOE. Some of the key drivers of mRNA yield were buffer constituents, specifically using EP sodium hydroxide and magnesium acetate instead of magnesium chloride. In addition, NTPs being at a concentration of 10 to 15 millimolar and having the pH at 7.6 to 7.9 were associated with reaching the theoretical maximum of mRNA yield while maintaining high mRNA quality. So to conclude, depending on what level your drug product is in, will dictate raw material process flexibility and documentation requirements. At the discovery phase, the process is at the microliter to microgram scale, and raw material selection has a lot of flexibility. This is where foundational DNA template considerations and mRNA synthesis process parameters are determined. At the process development phase, the scale is now at the milliliter and milligram scale, and raw material selection considerations should focus more on quality, as your IVT is getting closer to process lock. At this phase, the process is being optimized further through DOEs and process development runs. But in the manufacturing phase, the process should be locked and fully reproducible. Key performance indicators are already established from the process development phase that guided when the process parameters and raw materials selected reached the acceptance criteria needed to manufacture RNA drug product at scale. GMP raw materials, with all essential regulatory support, are required at this phase of drug manufacturing. Finally, Thermo Fisher Scientific offers a GMP portfolio that has all the critical quality attributes discussed in this presentation. We provide value that goes beyond the product in terms of quality and experience in the mRNA synthesis space. Our products provide consistency and reliability to your process with all the documentation needed. When working with Thermo Fisher Scientific, it is a partnership above all else, with our mission being to act as a secure and preferred supplier for in vitro transcription. We are happy to provide our customers proven peace of mind as they develop mRNA therapeutics and vaccines for the future. If you have any questions, please contact me and I will assist you with your inquiry. In addition, we have a website shown in the slide at thermofisher.com/therapure, where you can learn more about our product portfolio and mindset towards raw material quality. So thank you for your time, and do you have any questions?
Thank you for that, Daniel. I'm sure our audience thoroughly enjoyed your presentation just as much as I did. As with the other talks in this webinar, we're likely to receive a lot of questions, so I'll move straight onto this now. We will answer as many questions as we have time available for. Your first question today is, how can I make sure I get the same quality materials when I scale up?
That's a great question, and I assume the question is for raw materials. And in that case, check with your suppliers or vendors as you start looking for products or for process development. They should be fully set up to supply you with secure and scalable materials throughout your process. A vendor should have multiple sites across the globe with built-in supply chain redundancy. They should have the proven experience supplying these materials at scale to customers with minimal interruption, even at times like the pandemic. So to sum up, look for a supplier that offers redundancy and reliability even in tough times.
That's a fantastic answer. Thank you for that, Daniel. Moving on swiftly, your next question is, what are some of the differences with IVT when it is done at small versus large scale? What changes?
Small scale, mid-scale, and large-scale IVT differ in their process and are not 100% scalable in terms of product quantity. At small scale, IVT is usually done in a test tube to 20 microliters to maybe a mil, and thermal mixing is usually enough to run that reaction. But at mid-scale, it is usually when small-scale bioreactors are incorporated into the process. The advantage of bioreactors is the level of control you have over the process. With that said, it requires additional parameter optimization for temperature and agitation, because it usually uses a rotor, pH, buffer type and composition, NTP concentration, magnesium, and other IVT reaction components. And finally, the same parameter optimization at mid-scale should be done at larger scale. Larger scale processes require materials to be of GMP quality, so that is when raw material quality considerations are paramount.
Thank you for that. That is excellent. We have time for one final question today, which is, should I test my process by DOE for each new mRNA sequence?
That's a really good question. And we highly recommend that testing each mRNA sequence should be done because the DNA template and the sequence itself could impact your yield. Some factors that could affect mRNA yield and quality as it relates to the DNA template and mRNA sequence are like your sequence secondary structures, the composition of nucleotides that are in the sequence, the length of that sequence, the actual composition of the promoter and how that affects the DNA template, or how that affects the T7 polymerase as it goes across, and your type of DNA template, whether or not the sequence needs to be A-tailed or transcriptional capping sequence, and other factors that are specific to these sequence modalities. And because of their potential effects on IVD performance, it's recommended to test each sequence in a DOE.
Thank you so much, Daniel. That is unfortunately all that we have time for, but thank you for taking the time to present with us today.